everyone. Thank you for attending. I'm Kelsey Bisker, and today I'm going to talk about my research on the effects of differences in duration of larval period upon the field performance of cultured eastern oysters. So first I'm going to go over the eastern oyster and the associated oyster aquaculture, and then some background research, and then the reason for this research, my objectives, and the 3 stages of oyster production with the associated results, and then finally conclusions. So the eastern oyster, or Crassostrea virginica, is in the mollusca phylum and part of the Ostridae family. These oysters are cupped oysters, which is a trait that oyster farmers will take advantage of to create deeper cups to sell to the half-shell market. These oysters range off the coasts of Canada all the way through the Gulf of Mexico. They are found in brackish to salty waters and have been found to tolerate salinity from 5 to 40 parts per thousand, but for physiological growth, the optimal salinity is 15 to 18 parts per thousand. When the water temperature reaches about 25 degrees Celsius, this is when the oysters will begin to reproduce. They are broadcast spawners, which means that they will release their gametes into the water column and fertilization occurs there. So oyster aquaculture is continuing to increase throughout the nation along the western, eastern, and Gulf coasts. Nationally, oyster production is valued at over $185 million and accounts for 47% of the marine aquaculture production. Here in the northern Gulf of Mexico, oyster farming began in 2010 with the first oyster farms in Alabama and Louisiana, and now there are more than 100 oyster farms located along the states that border the Gulf Coast. So with the increase in oyster farms creates an increase in seed demands, which is very important for hatcheries to understand how larval performance affects the field performance of these oysters, because ultimately larval performance can impact commercial hatchery production as well as farming yields. So larval experience has been examined in many aquatic invertebrates, such as that invertebrate larvae have found to differ greatly within the same culture in terms of survivorship and growth rates, whereas the larvae will reach the pedivilager stage at different times compared to their siblings. And these variations have been examined in both the hard clam as well as the eastern oyster, where both studies have found that the larger larvae continue to have that better performance in the juvenile stage compared to their slow-growing siblings. And lastly, marine species have been found to postpone their metamorphic phase because of a stressor that occurs in the environment, which creates a latent effect that affects performance in their juvenile and adult stages later on. So the reason for this study is that farmers have began to question hatcheries on the quality of the seed that they're paying full price for. They're wanting to know if the seed they're receiving are bad seed, or if the seed were slow-growing larvae that were the last ones to be put to set to complete metamorphosis, and if that performance will then show up in the field. So I like to think of it as, are they the runts of the litter, and will they eventually be able to catch up to their larger siblings? So for my objectives, I wanted to determine if larval duration period affects metamorphic success, performance in land-based nursery culture, and performance in the field grow-out culture. So to begin, on July 10th, 2018, at the Auburn University Shellfish Lab, we spawned diploid oysters from 2 distinct broodstock lines. 37 females mated with 13 males, and we got a 91% fertilization rate. Tanks were stocked at 10 fertilized eggs per milliliter, and throughout the culture, normal hatchery procedures were conducted to be able to mimic a commercial hatchery, since oyster farmers will be purchasing their seed from them. So larvae were fed a shellfish concentrate diet, and drain downs occurred every other day until day 8 of culture, where they were performed daily thereafter. So this is just a graph to illustrate the change in shell height throughout the 11 days of larval culture. You can see that the variation begins around day 3 and just increases from there. So once when larvae started showing a foot, which is pictured in that bottom left corner, and have a dark eye spot, they are referred to as eyed larvae, or they have reached the petivillager stage, meaning that they are ready to be set to achieve metamorphosis. So we graded our larvae on a 200 micron screen in order to call off these eyed larvae to be put to set in our downwelling system. So to determine our groups, we based our number on the days that required the larvae to become eyed larvae. So the day that had more than or had 900,000 or more eyed larvae come to set, which was 11 days post-fertilization, we'd assign that as our early group. And the last day that had 900,000 or more eyed larvae come to set, we assigned that as our late group, which happened to be 18 days post-fertilization. All days were set in our downwelling system, and so we were able to determine determine our peak group, which we created this bell curve, and our peak group was the day that the most eyed larvae came to set, which happened to be 13 days post-fertilization. So at each day, the larvae retained on that 200 micron screen, the eyed larvae were stocked in 3 silos at 300,000 per silo in a downwelling system, and we gave them 4 days to complete metamorphosis. On that 4th day, the spat were graded on a 300 micron screen and then placed in our upwelling system. Within the upwelling system, we gave them 5 days so that we can then grade the seed on a 600 micron screen to account for the metamorphic retention success. So shell height was taken throughout this process, which is the maximum distance between the hinge and the tip of the new shell growth on the bill. So for our results, to begin, for all the analysis that determined if larval duration period has an effect on that x-axis, you will see the early group will be green, the peak will be blue, and late will be orange. And on our y-axis, for retention percent, in order to determine the success, we found the percentage of the oysters that successfully developed from the petivillager stage to an oyster seed, which is what we counted for for the amount of oysters that retained on that 600 micron screen. And you can see that there is no significant difference among the 3 groups. And then onto our growth rate, which is on the y-axis. This is actually the average change in shell height, but for the rest of this presentation, I will refer to it as growth rate, even though statistically I did not measure individual growth, but rather the differences between the average change in shell height of those silo replicates. And so for the 4 days that the oysters were given to complete metamorphosis, you see that there was no significant difference in that growth rate. So on to our land-based nursery culture. After oysters retained on that 600 micron screen, a series of grades were completed through the land-based nursery culture in order to keep the culture growing and to reach the size for field deployment. Oysters were graded to determine size class to be able to place on larger mesh silo screens to be able to allow greater flow rates and more phytoplankton available for consumption. So to ensure that we were being objective as possible and not calling out those smaller individuals that did not retain, we created a move up rule, which was that the grade was only completed if 75% or more of the total larval duration period retained on that next size up mesh grade screen. So if the group did not achieve this move up rule, then they were placed back into the system as their previous size and were given 2 more days until the grade was completed. Every grade densities were decreased to avoid overcrowding, And so the series of grades that we did were that 600 micron screen, and then a 1.4 millimeter, a 2 millimeter, a 4 millimeter, and then a 6 millimeter. And those were scheduled to be done every 7 days, but of course we abided by that rule. And then when groups were trained on that 6 millimeter sieve, they were deployed to our field nursery sites. Each group actually ended up being cultured in the land-based nursery system for a total of 37 days each. So on to our nursery results. After 5 days in that upwelling system, the growth rate between that 300 micron grade and the 600 micron grade, we find that the early group has the growth advantage for the beginning of that land-based culture. And then as we continue through our procedure, the growth rate between the 2 millimeter to 4 millimeter grade, we still see that the early group has that growth advantage by being significantly faster than that late group. However, after 7 days, when the oysters retained on the 6 millimeter mesh, we see that the peak group now has the growth advantage and the early group has slowed down. So larval duration does seem to have an effect during the land-based nursery culture. So onto our field grow out methods. Each group spent about 6 weeks growing in our field nursery field site. And so in order to be able to have that 75% or more retain on a 12 millimeter screen. So when we were confident that we reached that percentage, oysters were brought in October and were mechanically graded to determine that retainment success. And then each group were stocked at 400 oysters per basket and were deployed to 3 of our Auburn University field sites. The leftover, and they were combined, so each group, like the early group was combined all together to be able to make up these 400 oysters per replicate. And the leftover oysters from each of the 3 groups were split among 4 commercial farmers evenly so that we could see how the performance, how the different groups performed at their different farm sites using different techniques. Samples were completed in February, May, and August. And our on our February sampling site day, we decreased the oysters per basket to 100 oysters. And then in our May and August sampling dates, we took live and did counts of those oysters. And at each of the sampling times, we took 30 subsamples to complete our measurements. And so this bottom right picture is an example of one of our response variables, which was condition index, where we took the whole wet weight of oyster, weighed that out. Then we shucked it. We separated the tissue from the shell, allowed them to completely dry out. And then we took the dry shell weight and the dry tissue weight to be able to determine the meat to shell ratio. So this is an illustration of where the oysters for each group were deployed. The circles represent the 4 commercial farm sites, and then the stars represent the Auburn University field site. So the purple is our Grand Bay Oyster Park, the red is our Bayou Sullivan Oyster Park, and the orange is our Auburn University Shellfish Dock. So for this analysis for our field grow out variables, we set field site as a random factor because these were all mid-salinity sites, and so we just wanted them to be as a representation of just farm sites across the northern Gulf of Mexico and represent certain conditions the growers will experience at their own farm sites. So for the grow-up gear that we used, the oysters were grown in 6 replicate B.Sc hexagonal baskets per treatment group, which were arranged randomly along the adjustable longline system. So the top picture is just the arrangement, and the bottom picture is the adjustable longline system with an example of a basket. and the line was placed at an intertidal height at each site. Shell height, shell length, and shell width measurements were also taken of those subsamples of oysters. So for our field grow out results, first we will look at the effects of sample time, where the treatment groups and field sites are averaged together for each time point for this analysis. So on the x-axis will be sample time, and February is a blue-gray, May is the turquoise, and August is that light blue. And for our growth rate, we see that there's no significant difference, meaning that they are have a consistent growth rate over time. So for our dry shell weight average, there is a significant difference between February and August, meaning that these oysters are getting heavier and growing throughout the sample period. And so for our average condition index, February is a winter month, and these are diploid oysters, so they have a higher meat condition. However, when the water temperature warms up in May and August, the oysters will be spawning, so the condition significantly will decrease in May and August. And then for our cup ratio, we see another significant difference between February and August, which just means that the oysters are putting on new shell growth and are tumbling throughout the field grow out to create that deeper cup ratio that farmers really like. So this change over time just concludes that the oysters are growing at our 3 farm sites and just represent good conditions for those farmers. So now to review the effects of larval duration period on each group. The groups include the averages from the field sites and sample times to show this effect. And there's no effect on growth rate, no effect on dry shell weight, no effect on condition index, and no effect on cup ratio. So looking at those differences between the larval duration period, there is no effect between the 3 groups for these responses. And to determine if there was any differences in survival between the 3 groups, we multiplied the survival percentage from May and August together to determine a cumulative survival, or the chance of surviving to the end of the sample time, which was August. Since each time we took 30 oysters from each replicate, so we're just accounting for those. And we found that there was no difference in survival. So overall, larval duration period has no effect. However, one of the commercial farm sites did find an effect on growth rate. The late group has that disadvantage and is has a significantly slower growth rate. And this site also has a substantially lower growth rate compared to the Auburn University Shellfish Labs field sites. So this site is actually located just south of a freshwater input in the Mobile Bay. So salinity gets pretty low throughout the year there. And also, according to the farmer, there's a potential for low dissolved oxygen at times. So this farm site is actually a very stressful environment to grow oysters in. So as a conclusion, we found that there was no significant effect of larval duration period on metamorphic success. At first, we saw differences between the larval duration period in that nursery culture. However, the performance among the 3 groups showed no differences in response variables during the field grow out. But future work does need to be done to determine if growing depends on the conditions at each site, such as if stressful environments affect it, such as that farm site, that commercial farm site that we saw that effect in. And then additionally, it is important to consider other factors in larval production that might affect subsequent performance in order to support the growing oyster industry, such as salinity and nutrient stress in that larval production. And I just want to thank Dr. Bill Walton, my advisor, and my committee members for helping me out with all my questions, and as well as the whole Auburn University Shellfish Lab team for guiding me through this project and assisting me with everything, and from the hatchery to the field, and then also the farmers who dedicated their time towards raising my seed. And with that, I will take any questions.